Now it's my pleasure to introduce today's speakers, Dr. John Mumford and Dr. Michael Bonsall. John is Professor of Natural Resource Management at the Center for Environmental Policy at Imperial College London. He's an entomologist with experience of area-wide pest management and leads the non-native species risk analysis panel in Great Britain. He's also been a contributor to guidance on environmental risk assessment with the World Health Organization and the European Food Safety Authority, and leads the internal risk analysis work of the Target Malaria Project of the Bill and Melinda Gates Foundation. Michael is Professor of Mathematical Biology at the University of Oxford. He leads a research group on mathematical biology that focuses on integrating theoretical and empirical problems in the life sciences. Particular themes of research in the group range from problems in mammalian developmental biology and stem cell dynamics, ecology and evolutionary biology, GM insects, the dynamics of communicable disease, and also non-communicable psychiatric disorders such as bipolar disorder and PTSD. He has worked at the interface of policy and science around GM insect releases, serving on the UK DEFRA Committee for Releases to the Environment, and working with the House of Lords Science and Technology Committee, the European Food Safety Authority, and the World Health Organization. With that, let me say welcome, John and Mike. Thank you for your willingness to share your thoughts with us today. And so John's going to begin. And so John, let me invite you to start with your part of the presentation. Please go ahead and take over, and I'll let you know when we can see your slides and we can hear you.
Okay, thanks very much for the introduction, Hector. I hope this is coming through clear now to everyone.
It's coming through just fine.
Good. I'm going to introduce the rationale for the use of models in environmental risk assessment for gene-drive insects. And then Mike will come in later in the presentation to present some examples. And we're very conscious of the fact that we've got an audience here that includes some people who are very familiar with models and some people who are very familiar with environmental risk assessment, and some both. And so we're trying to speak to everybody as we go through this presentation. You've heard in the introduction from Hector some of the experience that Mike and I have had, particularly in contributing to guidance from international organizations on environmental risk assessment for GM and gene-drive insects. And we've been very conscious while we've been doing that of the need to think about what it means for the developer or the applicant to actually deliver what's needed to meet regulatory approval. So we're trying to make sure that the guidance that we've given over the years has been practical, as well as meeting the theory and concepts of regulatory approval. So we're going to talk here about why and how models are used and give you those a few examples. And we also are looking forward to greater detail by some of the other speakers that are coming up in the next 3 weeks. So let's move on here to what's a commonly expressed concern about gene drive. And in fact, one of the arguments about why it might not even be possible to do risk assessments for gene drive. And we would argue that it is not really true that anything can happen. There's really a limited set of things that can happen that we're actually concerned about. And given that's the case, then the second half of this statement that we can't observe, review, or model everything is really not very relevant. We should recognize that an environmental risk assessment is going to occur well into the development of a gene-drive product. And so there are already at that stage going to be quite significant expectations and assumptions from the applicant or the developer about what they expect it's going to do, how it's going to behave. And those form the basis for a risk assessment. It's up to the applicant to present what's going to happen and then what could deviate from that. So that narrows the target considerably for modeling or indeed for observations and review. So if we look at the things that can happen and that are related to protection goals, they really fall into a fairly narrow set of categories here. There are direct target organism effects, so things like the gene frequency, the population density, and the gene products, proteins, toxins, for instance, that could be produced by the target organism. And then there are indirect non-target organism effects, or NTO effects, which are largely related to density-dependent relationships between the target organism and other non-target organisms, or to a dose response to gene products within the target organism, or to changes in the target organism that might affect its ability, for instance, to transmit pathogens, or to niche replacement by new non-target organisms. And we may also have specified in regulations abiotic effects that have protection goals, such as water and soil quality. And these may also then be related to the gene products or to the densities of target or non-target organisms. And there is a time scale that's important, apart from the protection goals. So there are the qualities that we are trying to protect, but there are also time periods and ultimately spatial scales over which we want those protection goals to be achieved. And when you look at the time scale, we feel that that breaks down into a transition stage as the gene drive starts to work, and then moves on to some longer term equilibrium. So overall, as we think about the things that we need to model, we've got the relatively few protection goals and two temporal and spatial scales, the transition and long-term equilibrium, that we need to take into account. And so again, we've narrowed the target very considerably for models. Now, it's important that we think about some definitions here, and this is going to be in relation to risk assessment, to models, and to some of the components of those things. So the first thing we need to be aware of is what environmental risk assessment is about. And so it's around the likelihood of a series of events or conditions or relationships that result in harm and the magnitude of that harm, and specifically harm to the environment and human health. So quite focused issues of assessment there. So it's about harm to the environment and human health. And the risk assessment will identify concerns, it'll characterize hazards, exposures, and distributions, and then consider management options and overall risk. And models in particular are often seen as focusing on the characterization or quantification of the hazards, exposure, and their distributions. But they also help to articulate concerns, for instance. So in identifying concerns, we're able to express them in the terms that would be appropriate to models. So what goes in, what comes out, and what are the processes that could make that happen. And models are recognized in various regulations, and we'll mention throughout this presentation some examples from European regulations, but there are similar ones that will exist in other jurisdictions as well. But in the structuring of a risk assessment, so in the problem formulation stage, the current EU directive on the subject mentions mathematical modeling as one of the three tools that are particularly relevant to structuring and conducting the assessment. So evidence from previous experience or observations, published data, and then mathematical modeling itself. So a model, to define that, is a representation of relationships in nature. And it is determined by the modeler, but the modeler should reflect the interests of the regulators in society in the way that the model is structured and the way that the outputs are presented. And in terms of the outputs that are relevant for an environmental risk assessment, what we're looking to do is to predict outcome distributions within the limits of the available data. So the data may have a limited range and there may be a great deal of uncertainty. So that can limit the way that we can predict outcome distributions. It's generally an assumption that there is going to be some sort of stable equilibrium that develops in the short to midterm, and that would imply longer stability. So again, from the point of view of defining the limits of our models, we don't have to model to infinity. We have to model till we are convinced that there will be a relatively stable equilibrium. And the models may have various types from conceptual through to quantitative. We can also see that models have a form of taxonomy. There are ranges of models that range from general to specific, from real to precise, and so on. And for an environmental risk assessment, we're really looking at this right-hand side. Most of the guidance around the assessment of gene drives is promoting a case-by-case approach. The case-by-case approach argues against the general, and the parameterization of models is very important in those specific cases. So it's on this, this line from realism to precision that we would expect to see the sorts of models that would be relevant for environmental risk assessment. Now, environmental risk assessment is about harm. So we need to define harm. And so harm, as we are defining it here, is an adverse deviation from a reference outcome. And adverse and reference are both subjective. So we're setting up within an environmental risk assessment process using models some sort of technical evidence-driven process to determine what the risk is and to think about risk management that might be necessary for it. But we should recognize that at the very core of the risk assessment, we have subjective elements that are related to what the reference outcome is, and indeed to what people are concerned about, what the regulators or society are concerned about. So it's a combination of a technical process, but with that subjective element built into it. And ultimately, the models are looking at event to harm chains. So they're looking at an event, the release of the gene drive organism, the conditions of the receiving environment into which it's released, the processes by which it spreads and persists, the outcomes that occur, and then why those outcomes produce harm. And if we put that harm in the context of models, we need to understand that the environmental risk assessment can only assess plausible event to harm chains. If you cannot express it in an event to harm chain, it's not possible to do a risk assessment. And models are very good mechanisms for defining those chains, and they're essential for looking at dynamic impacts because of the evolving nature of the relationships between the target organisms and the non-target organisms and the various impacts that could occur. And it's meant to give some quantification of harm, but the quantification of harm is going to be in relation to the reference, the acceptable reference outcome. And so that's going to be dependent on the comparators that we select, structure of the model, and the available data. Now, another thing that has come up with gene drives on a regular basis is that, well, they're new, and that therefore there is some novelty in the features of gene drive that could lead to novel harms or novel pathways to harm. And a question that we need to consider, and which we can discuss at the end of the presentation, is whether novel features lead to pathways to harm that are new to modeling. can we model something that's novel? Is there anything so new that it's beyond our experience or our capability? And we should recognize that models are not expected to predict the exact outcome. They are expected to tell us something about the likelihood of an acceptable level of harm being exceeded. So we can use an analogy from weather prediction. We're not trying to predict the exact amount of rainfall that will occur tomorrow. We're trying to predict whether it's necessary to take an umbrella. And there's a technical element to that about the level of rainfall, and there's a subjective element to it, which is my tolerance to getting wet. So it's a very different kind of modeling than predicting the exact amount of harm. So the sensitivity of models around that acceptable level of harm is very relevant. And the tighter the acceptable level of harm is defined, or the lower the level is, that can affect the sensitivity of models at that point. So I want to go on to talk here now about efficacy and harm. And to start with the observation that traditionally environmental risk assessments separate risk from any kind of measure or consideration of efficacy or performance. Those kinds of evaluations are, or have been in the past, generally seen as being related to benefit analyses or to the decision-making process around an implementation decision. So we would try to say the risk assessment is only about harm, and efficacy and performance are around benefits. And in fact, if you go back to the original European GMO directive from 2001, so 20 years ago, there's no mention of efficacy in relation to risk assessment in that directive. As we move to gene drive systems, though, the whole way in which they work is around population effects, and those population effects are clearly related to efficacy, and they affect the nature of harm. So efficacy, for instance, would reduce the target organism, and that causes indirect effects on density-dependent non-target organisms. It affects the transmission of pathogens and the frequency of mutations and the impact of the gene products that might be toxic. And there's also considerable concern expressed by regulators and the public about the loss of efficacy and seeing resistance developing from a control measure resulting in resurgence as itself being a source of harm. And what this implies is that in the course of a risk assessment, or over the time period that a risk assessment is being considered, that we have new reference levels. So we've got the current reference level, and then as the equilibrium sets in and control takes place and resistance may occur, we're implying a new reference level where harm is set against that higher level of control that might exist in the future. So looking at some specific examples of this relationship between efficacy and harm, within the Target Malaria Project, we've recently conducted an analysis of pathways to potential harm for a specific gene drive strain. And an article on this will be coming out in a couple of weeks, certainly by the end of this series on modeling. The pathway numbers in that analysis are not particularly relevant. You could get slightly different numbers, or maybe much different numbers with different strains and with different structure of analysis. But in this particular one, we had 46 pathways. And in terms of the analysis steps that would be necessary to evaluate or assess those pathways, 35 out of the 46 included some use of modeling. And of all of the analysis steps across the 46 pathways, 58 of them, of the 204 that we looked at in the project, involved modeling of some form. And within those, 60% of the analysis steps that involved modeling were around issues to do with efficacy, so the net effect of harm following successful suppression, or trying to estimate what the break-even effect would be from particular functions within pathways to harm. So I just use this as an illustration of the fact that we're recognizing as we start to do very detailed pathway to harm analyses that modeling is an essential step in resolving them. So regulations on ERA now expect models. We saw that in the current European directive on environmental risk assessment from 2018. But going back over the last decade, EFSA recommendations in 2013, the World Health Organization ones in 2014, right through to the EFSA opinion that Mike and I were involved in last year on gene drive guidance for environmental risk assessment, they are all recommending models. And the demand for modeling is seen to be increasing over that time. So modeling is a key word in the EFSA 2020 evaluation, but not a key word in some of the earlier guidance documents that came out. There's an expectation in these recent documents of a greater dependence on models. And I think that's illustrated by the analysis that I showed you a moment ago from the Target Malaria Project. That modeling, while we are going to depend on it, modeling is not the only answer to an environmental risk assessment. So we can use models together with observations and data from the literature in a weight of evidence approach that together will actually describe the risks that we're trying to assess. And another thing that comes out in this demand for models is that there's a greater demand or emphasis on scenarios and the assumptions around those scenarios that are relevant to gene drive. So we are assessing scenarios that relate to event harm chains. So I mentioned earlier the issue of novelty. So what's novel about gene drives that would be relevant in an environmental risk assessment and in a model that we would use? Well, in the EFSA 2020 opinion, the novelty of gene drives came down to a relatively small number of conditions. There was inheritance and persistence of traits, and there was a large spatial and a long temporal scale. And those were the main features of gene drive in an ecological sense that were novel compared to some of the previous GM controls. Now, we would assume that a practical gene drive system that would be put into effect would be likely to have a relatively high inheritance rate and a fairly rapid spread in the environment in order to achieve its goals. And so when you look at the sorts of models that are produced, and we've got a few examples here on the right, some outputs from them. You see in the ones to the left, the column to the left there of graphs in Beaton et al.'s modeling, you get over the course of 20 to 30 generations, you get a transition from the original state through to an equilibrium. And on the right in the stochastic models, which I'll take some examples here from North et al. They're looking at environmental qualities in space, fairly limited environmental qualities in a series of spatial connections, and seeing how those relationships would develop. But we're looking at relatively short time periods and at localized space as the change takes place. And that's quite important, I think, to think about that we're not modeling everything for all time. We're modeling the bits of everything for the times when something's happening. And that narrows down the spatial and temporal context of modeling considerably. So here we see another representation of spread in a model by Beaton and some others. And it represents a snapshot of a wave of spread into a population of a genetic component. And so what you see in front of the wave to the right of the figure is the unaffected region. You see to the left behind the transition wave the new equilibrium, which may be elimination or suppression at some level. In this case, it's elimination. And then you've got a 30 or so kilometer wide wave of transition zone in this particular model. And what we're really following here is three stages, three states. We've got the status quo, we've got the transition wave moving through space and time, and then an equilibrium behind. And those are the three stages that we have to model. And something that we have to ask ourselves here then is, so what is the reasonable time frame to consider? Do we expect a single model to cover the whole of the time, or do we have a model for transition, which occupies a relatively short period of time and a fairly localized bit of space, and then an equilibrium stage model that covers a unit of equilibrium time and example spaces? So again, we're not trying to model absolutely everything all the time forever. And another thing that we have to consider in such models is how in the model output we value the transition stage and the risks that occur during it to those that occur during the elimination stage. And we'll go on to show in a moment some examples that show how different the risks are during the transition compared to the equilibrium that comes later on. So thinking about this novelty, we are still dealing with basic ecological principles, which have been modeled for 100 years. We're looking at interactions between target organisms and non-targets. We're looking at density-dependent relationships. We can validate and test some of the assumptions of models because there are some natural systems like Wolbachia that are similar to gene drive in many respects. And there are other management processes like biological control, which also have many similarities to the relationships that we would expect to see in gene drive in terms of spatial and temporal spread. So we think that models can predict gene drive performance, and they can predict, based on pretty well-known principles, how organisms will react with each other. So what kind of effects are we likely to see? Well, and particularly ones related to efficacy and its relationship to harm. And they really fall into two categories here as we see it. So there's a harm that's dependent on the density of the transgenic population, and that harm would come because the transgenic population is too high in order to produce harm. And that could be that it's very high during the transition wave. So there's a lot of transgenic organisms at that stage, a high density of them. Or in the equilibrium dynamics, it still may be too high in some cases. And the sort of effects that we're thinking about here are transmission due pathogens, or it could be the direct effects of gene products that are toxic in the environment. So those are related to the density of the transgenic population. Another kind of harm that's related to efficacy is the harm that arises out of the density of the overall target population, so including the residual wild population as well as transgenics. And that harm comes from that population being too low. So this is around the dynamics of non-target organisms that are dependent on target organisms. And the question here really is the target organism a key species in the environment that's going to affect non-target organisms that are valued and provide ecosystem services? There are also some density independent effects which would be unrelated to the efficacy of the system. And an example of that might be very low threshold initiation of resistance, which is not very closely related to the density of the target organism. The structure and interpretation of models is quite important, and there are several different levels of comparators that models could consider. And we would argue that really for an overall environmental risk assessment, we're going to need to be looking at all three of these levels. So individual comparators are going to compare fitness. Population effects are important, particularly because they relate to efficacy. And the systems effects are important because of the knock-on effects to management and some of the consequences of management being withdrawn because of the effects of gene drive. So a few points now about using models. And we look ahead here to Kim Pepin and Andrew Goldner's presentation in a couple of weeks, where they've recently presented a paper on some of the issues around models that we very much welcome and look forward to hearing in more detail in a couple of weeks. The need for consistent model structure, both between analyses and also within a series of analyses in a single risk assessment. There are issues around interpretation, and something I think that we're all aware of here now with the COVID pandemic is the massive increase in awareness among the public of the role of models in ecological, epidemiological management. And this may be a good thing in terms of risk assessment for gene drive, that there is a greater awareness of the public of how models work. But we've also seen within the COVID pandemic considerable public misunderstanding and distrust of models. And so that's something that's going to need to be addressed. And there's also a role for models to structure data collection. And so we'll hear more about this in 2 weeks' time. There are various uses for models at different stages. So in gene drive design, informing risk hypotheses, right through to post-release monitoring. And as I've just mentioned, in this essential need for communication throughout, that models are a way of providing a vehicle for that communication. A number of purposes, and these are fairly evident. You could have models that are built specifically for a risk assessment, or you could have a performance model that is secondarily applied to a risk assessment. Goals, we'll look at a few later on that could go from statistical measures like power estimation, sensitivity analysis, and so on. The key point is that the models need to be designed for a particular goal and that they are clear in the assumptions that are used in them. And we need to recognize the limits of models. As I described earlier in Levin's triangle on models, that we are looking at this side between realism and precision. We must have data that allows us to parameterize models. A risk assessment must be built on some characterization of hazard and exposure, which requires data to parameterize it. And there are a number of issues around how we validate models, especially prior to release. And these are something that need to be kept in mind, and I expect we will see examples of that over the next couple of weeks. Many of the guidance documents that have come out over the last decade have highlighted the need for more guidance on the interpretation of models that allow that validation to take place. We think also there's a role for models in evaluating the risk assessment process itself, that having models in a risk assessment process helps us to evaluate the various principles that come out in such evaluation frameworks. And we've illustrated it here with one from Kuzma, as produced in 2019, as a series of principles, humility, inclusion, and so on here. And by articulating the risk assessment issues in models, it helps to ensure that there is transparency, that there's a mechanism for discussion with external parties, there's a consistency, there's an ability to challenge, there's an ability to introduce new information or parameters or values later in the process. So the structure of a model helps, I think, in all of these principles in this particular evaluation framework, and there are various others that could be applied as well, all of which I'm sure would also benefit from models. So let's quickly look through the use of models in the various stages of research and development. So in cages, so the very early stages of testing in confinement, there are statistical models largely around fitness, and they help us to develop hypotheses for field releases. In field releases, we've got models that help us to estimate populations, models that help us to determine the impacts of specific parameters, such as vector competence or vectorial capacity. Some of these models could be based on cage data. Some of them, like the lower one on the right that is illustrated from the CSIRO, looks at expert opinion and uses that to set prior expectations. And the field release models can move on to ultimately to quite complex dynamic models with networks of interactions. We could go to deployment models, which are largely going to be network models working on a large scale that take into account current control actions and the release scenarios and help to make decisions about implementation. And finally, post-release monitoring models, which are going to determine the statistical power of a sampling regime. So we've dealt with novelty. We've talked about uses. Let's just think quickly about space and time. And how's that dealt with in models? And looking at the various models that are out there for gene drive, we're seeing very significant range in spatial coverage. So from very localized models up to 1 million square kilometers in the North et al. Models. We're looking at time ranges from tens of generations up to several hundred generations. Timescales generally in the range of 8 to 16 years. So again, both the spatial and the temporal limitations on models are they're coalescing around these sorts of scales as far as we can see. Are there limits on the spatial scale? Obviously, the bigger you make it, the greater the heterogeneity of the receiving environment. And that creates problems in just operationality of a very large scale model operating over a very long time. An alternative to doing that is to break down the risk that we're looking at into smaller spaces and shorter times. So we looked, for instance, earlier at the wave period and the equilibrium period. You can use short time frames there. And we could take representative receiving environments and say, during those time periods, in those kinds of receiving environments, what kind of harms could develop. And so that may be a more practical way of dealing with the large time and space of something that moves out and lasts more or less forever. We've seen in some of the modeling that's been produced, here's some examples taken from North et al. That they found that the receiving environment, for instance, is critical to spread and dynamics. It is, in this particular model, the heterogeneity in the receiving environment is more significant as an effector than different levels of fitness. And we can see on the left in the two squares here on the left, which cover 1 million square kilometers centered on Burkina Faso for the release of a particular gene drive organism, that the levels of suppression, so red is higher levels of suppression, blue is lower levels of suppression in the efficacy model, that in the red areas, we would be expecting more of the dependent NTO risk. So the risk there is going to come from the target organism being at a low level and affecting non-target organisms that way. In the blue areas, where there's less efficacy, we've got more residual transgenic target organism density. And so the risk is going to come from more from the effect of, for instance, a gene product that might be present in that higher density of target organism. So the receiving environment variability affects efficacy, which determines then which kind of risk we're likely to be affected by. So we're now going to go over to model examples, and Mike will take us through just a few of those.
Great. Thanks, John. Hopefully everybody can hear me okay.
Yes.
Hopefully everybody can see me too. Camera's on. So on the, just going to go through some examples on this first set of examples, really thinking about how we might address the ideas around transition from the transition stage towards what we think about a long-term equilibrium outcome. So on the animation on the left, and this is really just a cartoon about self-limiting releases, is on the y-axis you have the sort of potential of the system to change. So sort of high potential or low potential. So low potential being towards the bottom of that axis, high potential for change at the top of that axis. And when we're thinking about doing these sorts of control, this transition, we're going to start with these. When we're thinking about obviously mosquito control, we're thinking about how vector abundance being at some pre-control level, and then as we do our releases, so like either if that's a self-limiting release or a gene drive release, it's going to change the nature of the system. So one of the things that these models are able to highlight is the fact that any environmental risk assessment needs to be dynamic. That we start as we proceed through these releases, as we proceed through this, you can see the system changes. The propensity of the system to change alters as we do more releases, as that transgenic technology spreads out into the population and then in time. So we see the system collapsing in some sense from what was initially a long-term pre-control equilibrium. And in this case, what happens towards the very end of this simulation is that you see in the very bottom left-hand corner of the graph that the system just gets sucked into a stable state where there's low potential for the system to change and there's low vector abundance. And obviously that's an ideal outcome. But what this really emphasizes, at least in our minds, is that it is just it allows us to evaluate this idea of this transition, these ideas of transiting through different sets of dynamics, which are really important in terms of thinking about how we interpret that in terms of our risk assessments through time and space. The picture on the side, just to re-emphasize some of the things that John has already mentioned, is that we're not modeling exacts, we're modeling probabilistic outcomes. And the picture on the left, or the picture on the right, just shows stochastic simulations from a self-limiting release. So it starts at some point. So again, we've got population density on the y-axis through time on the x-axis. And what we're looking at is realizations, lots of model realizations, lots of different outcomes of the same model to stochastic change. And what this really emphasizes is that our bounds, we don't see all possible outcomes. We see actually the population declines, and it reaches in this case some sort of steady state where some sort of long-term a sort of equilibrium state. And in sort of population dynamic terms, that's really has some sort of bounds around it. So it's not a fixed point. There is some sort of spread around that. So we might expect that to be able to use these stochastic models in our risk assessments to sort of like to link outcomes with aspects of harm. So you could really think that you could put a horizontal line across that graph, where above a particular density of mosquitoes or control agent, that actually that would be unacceptable. And you could then work out from these sorts of models what the probability of being over that sort of unacceptable level would be. So with just some examples about those, just initial examples about how we can build those models, and just again to re-emphasize, we're really thinking about these along that precision realism axis of that Levin's framework, and really thinking about how they would be parameterized. John, can we have the next one, please? So this is just, again, John's already introduced this. This is work by Ace North, Austin Burt, and Charles Godfrey, modeling the sort of anopheles in villages at 1 million kilometre square scale around Burkina Faso. And again, what's really cool about this particular piece of work is that it shows two things. One, it shows there's nothing necessarily new in that, as is already emphasized, we can build in all of our understanding, our ecological understanding, ecological principles and sort of evolutionary principles about inheritance of these sorts of genetic constructs. We can also think that also understanding the sort of spatial and temporal extent, we can build this into our models. And as has been emphasized, the receiving environment becomes really important here. So the spatial map, as John has already outlined, just shows those villages across this 1 million square kilometre scale, with Burkina Faso stuck in the middle of that. And as we move through this, we can sort of like, as you step through this, you'll see that we see this sort of changes from high season to low season. This is all pre-control. So really showing that actually we can use our models to characterize the spatial and temporal extent of the receiving environment, that actually seasonality matters here, and that the way that we expect that gene drive construct to work is really dependent on understanding that receiving environment, and that is information that is characterized by this model. And it emphasizes again that we're using our models as representations. We're not trying to capture everything about this million square kilometers. Is actually being able to characterize those particular relevant aspects of the environment. And critical to this is the seasonal outcome. And what we see then is actually as we move forward, it sort of emphasize, the models emphasize again this set of transitions that we move from, where the densities of the transgenics and the target organisms alter through time. And we see that actually, obviously, there's a reduction. There's an orders of magnitude changes in the anopheles populations over time. They are seasonally dependent, and we get to understand again where our concerns might be focused to do with the abundance of the target as the target organism changes. So early on in control, there'll be lots of heterozygotes in time and space, and they may have that may focus our risk analysis and a environmental risk assessment at those particular points. There may be, as we move through control, we're seeing a reduction in the population size, and that again there may be residual concerns about the abundance of the transgenics and the transgenic organisms as that efficacy changes, and then we get to a point where there is low. So we've passed our critical threshold, which is demonstrated on the picture on the right. Where we've got numbers through time, we've passed that critical threshold. Well, now we've got sort of low target organism numbers, high efficacy, and the implications then oh, well, the risk assessment then might focus on an entomological, in this case, in this Anopheles case, might focus on an entomological outcome. It may be focused on an epidemiological outcome. It may be focused more on an ecological outcome to do with what is this relationship? How are these relationships between efficacy and harm changing as we move through these these controls, these stages of control? And these models are really highlighting the fact that this is a dynamic process. It really highlights that risk assessments should not necessarily be thought of as static things. That actually they evolve, that because the system is changing through time and space, our risk assessments should change through time and space. And it goes back to that sort of like the picture that John introduced from the Bateman modeling, which showed there's this wave of transition where there's something happening ahead of the wave, there's something happening behind the wave, there's something acting in the middle of that wave, and they all we need to be able to characterize our risk assessments according to those different aspects of the transition. And I think the final illustration I have is just to illustrate the sort of like how these sorts of, as efficacy changes, we can have impacts on non-target organisms. So this is sort of like a phase space spot, a space space plot. And in the middle, when it starts, in the middle of that, we start with a system in a pre-control state. So everything's at some stable state between your target organism and some non-target organisms. They say they're quite happily coexisting. And as we sort of run out our control, we see as our efficacy changes, as the impact of the transgenic on the target organism increases, and that's illustrated by the thing running forward on the very top, is that the efficacy is getting stronger. You're dynamically changing the interaction between that target organism and this non-target organism, and we see sort of, again, the expectation is that the system is going to collapse down, where if we're doing these sorts of suppression controls, suppression drives, is that the local elimination of the pest or vector leads to a shift in the interaction between non-targets. The other thing that this picture illustrates isn't just where the black dots are, but actually the different colors illustrate that actually the outcomes that could arise aren't necessarily fixed aren't necessarily fixed. Again, it's emphasizing the fact that we're estimating probabilistic outcomes, and the different colors highlight the different probabilities of outcomes. So the yellow bit being a sort of really high outcome, and the blue bit being a very unlikely outcome. So again, just emphasizing that actually those outcomes aren't exact, and they are probabilistic in their nature. So those are just a few sort of cartoons just to illustrate how we think that actually the models can be used in informing our sort of like thinking in our risk assessments, but also thinking about how we might use them in anger in sort of like parameterizations and explorations of particular technologies. I think we're almost at the hour, so I think in the sort of final set of points are really just to sort of emphasize the things that we've considered. Obviously, has been really, as lots of our guidance and opinions have highlighted over the last 5 or 6 years, that models are now to be expected in risk assessment. We shouldn't shy away from that, and actually they are going to be an integral part of us developing formal risk assessments for these gene drive type technologies. The novelty of gene drives is sort of like is less. Is there anything novel in this? I would argue, whether John supports me on this, but we'll see. But actually, in many ways, not. I don't. I think our ecological understanding is sort of our basic understanding of ecological principles and our basic understanding of gene spread is well developed in that Levin's framework we're way beyond the generality now. We understand this, so it is really understanding what is novel about gene drives, well, compared to other genetic technologies, is, as we've emphasized, the space and time aspects of this. So that was where we need to sort of like really think about how we can use our models to sort of like get a better handle on the risks associated with that. And that comes down to really thinking about how we link efficacy and harm. And as we've emphasized, I think that is really an integral part of this, that actually what our modeling is able to help us identify is that link, is how does that efficacy, how as efficacy changes, how does that introduce aspects of plausible aspects of harm that we would need to risk assess. We're not, as I said, emphasizing that models are, outputs are sort of like a probabilistic outcome and not an exact. Gives us ideas for being, again, characterizing when things become unacceptable and beyond a threshold. And I think sort of finally, I think with this is really thinking about the purposes of these sorts of tools in helping us develop risk assessments is really important. Where are we using them? Are we using them in our design of our constructs? Are we using them in understanding outcomes in small scale lab trials? Are we using them to sort of evaluate field outcomes? Are we using them in a sort of monitoring perspective, in a monitoring aspect? And I'm being super clear about that is really important. And as we hope we'll hear later in the series, the need for consistency of models is interesting, and sort of like whether we should have fixed frameworks for that everybody chooses to use for modeling the risk assessments associated with these technologies is something that we've just put out as a question, essentially, more than a statement of fact. I'm really interested to hear what this group thinks about how models should be used and how they should be, how we should build libraries of sort of like routines or functions, and how we should sort of bring the community along in thinking about that. And there's obviously some thinking that on this that we'll hear over the next 3 or 4 weeks, but also beyond that too. So with that, I think I'll stop. I know we've run over the hour or so, so I will pause at that point, and I guess we'll take questions. Thank you very much.
Well, thanks.
Thanks, Mike, and back to Hector.
Yeah, go ahead, John. Did you have something else to add?
Yes, I'm just thanking Mike and welcoming you.
Okay, thank you. And thanks to both of you, because this has been a great introduction to the whole series that we hope to have over the next 4 weeks. And actually, I wanted to just pick up on the last statement actually that you made, Michael, about the possible development of a library of routines, because it goes back to one of the first statements that John mentioned, and that is that we really still have to continue to consider things case by case, but perhaps there might be a set of generalized models that could be used, which then could be parameterized for specific cases. And I guess that's what you were getting at with that point about the library of routines.
Yeah, it's really thinking that can you develop that sort of library that would be adaptable for those case by case scenarios, or is it that you do have to? Is that just not feasible? And it'll be interesting to I think it's interesting to think about that, whether we can choose, given that in some respects, as we said, that ecological principles are well known and have been well known. Can we then just plug and play sort of thing in that? So in just building out models from these sorts of libraries of routines. Yeah.
Well, we've got quite a few questions that have been posted, so I'm going to get right to them. The first question is from Nikolai, and I guess the point he wants to make, well, I'm just going to read it, but I'm going to also interpret it. What's the actual reference of such a risk assessment? It seems the idea is to model the transition from one equilibrium pre-intervention to another post-intervention. However, predictions of climate-induced extinction rates in Africa and ecosystem transformations are already highly uncertain. Climate-attributed extinctions are now being reported all the time. So how can one model the effect and possible harm of a gene drive, essentially a non-event, against a rapidly changing background? I interpret this to mean how do the bounds of the background environment get incorporated into these models?
Okay, so I'll start on that one. Yeah, it is a very interesting question, and it raises the point very well that we've emphasized the dynamic process of the gene drive. Nikolai is emphasizing as well the dynamic process in the receiving environment. So what we've shown in some examples here is that the receiving environment and its heterogeneity is really important. I guess I would argue that gene drives are going to change a dynamic much faster than climate change. If it doesn't change faster than climate change, then it's not going to be worth introducing a gene drive, in my opinion. So the pace of change for gene drives will be faster than the pace of change from climate change. And I also think that the conditions of climate change many of them are already present across the range of climates at a continental level. And so we can already see how target organisms and non-target organisms are behaving in a range of climatic conditions. And the key question left is if climate change goes beyond any of those existing conditions. But if it's just moving a condition from one place to another, I think we can predict that, and we can deal with modeling gene drives in that change. Mike, do you have anything to add to that?
Yeah, just that I think also when we're building our models, we're really thinking about plausible pathways to harm. And I think the timescale over which they're operating, as John has emphasized, is really important, that actually we're really thinking that these sorts of interventions are going to happen over a few generations, a few years, rather than rolling out against rapid, against a sort of like maybe slowly or rapidly changing climate. Is important there. But I do think it's formulating these in such a way that they are direct. And if that's a concern, then yeah, we build that. We consider that within the frameworks that we have. And as I said, some of the illustrations have tried to get at that by thinking about that heterogeneity in the environment, looking at that how that receiving environments might change over this huge million square kilometer scale that ACE and colleagues have been working on. I think gives us a way in which we can incorporate that sort of information, seasonality, changes in seasonality, and asking against that the backdrop of climate change at whatever rate that is being expected to occur, how effective these things will be. But it really comes down to having, formulating the model in such a way that we've got, there is a plausible route to harm there that we are trying to understand. That's a good question. Thank you.
I just want to follow up on that, on the issue of timescales. John, in your part of the presentation, you mentioned that the timescales that modelers are using seem to coalesce to a time period of about less than 20 years. But we also hear about timescales of concern among people who are worried about gene drives, that the scales that need to be considered are essentially infinite. So what's the thinking in terms of limiting the scale to where that are being modeled now, the timescales that are being modeled now? Is that a limitation of the model or the modeler, or does that have some biological basis for that time frame?
I think it has a biological basis. There's a market basis and a biological basis. So the market basis, I would argue, is about the properties of the gene drive that anybody is likely to introduce for some purpose. It is most likely that gene drives will be expected to move fairly quickly. To introduce a gene drive and not expect it to have any beneficial impact for 100 years is unlikely. You're going to want it to work right now. So we expect the transition phase, which is one of the phases we think is interesting from a risk assessment point of view, to be taking place over a couple of seasons, a couple of years. And then after that, then the biological part comes in. We would expect a new equilibrium. Whatever happens is going to, it works or it doesn't work, or it half works. It's probably going to move to some new equilibrium. And by an equilibrium, I don't mean that it's absolutely not changing from day to day. There'll be ups and downs, but there will be some equilibrium that may involve cycles and so on, but it'll be relatively stable. And if you assume that there is going to be some biological stability, even with the potential upset of climate change and so on, that's also going to come in a reasonable number of generations and seasons and years. And so if you put together an assumption, and these are clearly assumptions, but you have to build assessments on assumptions that are transparent. The transparent assumption that we see is a fast transition phase over a couple of years, and then a fairly smooth move into some kind of equilibrium. And that could then be something in the order of 10 to 20 years. So it's not really surprising if you think about those assumptions of rapid transition and a fairly stable equilibrium, that's the time frame that modelers are looking at.
Interestingly enough, it seems as though those time frames don't differ much from what people are expecting to conduct risk assessments for non-drive organisms or transgenic organisms as well. It seems like that's been typically what they're thinking about.
Yeah, and it's also typical of modeling implementation decisions. Work that I've done over the years on implementation of SIT programs and the area-wide control programs. 15 years from an economic point of view it's a kind of comfortable compromise between the uncertainty in the future and the desire to project benefits out as far as you can. And you can't have it both ways. So that 15, maybe up to 20 years is about right there as well.
So I apologize for monopolizing the quick Q&A, but so I'll move down to a question by Nikki Faber, who says, thank you, first of all, for a great talk. Do you think we could and should use spatial models by using software like GIS? How can we include the ecological role of invasive species in an environmental risk assessment when a lot is still unknown?
Yeah, that's a good question. I think it comes down to the thinking about the choice of modeling and why you're actually doing that. So I think some of the examples we've presented have a sort of like a framework in the sense that we're modeling the sort of dynamics in terms of some ecological principles, whether that's through the sort of like release of these in a deterministic sense or release of these transgenics in terms of a stochastic sense or in terms of space. The other aspects of this that we sort of like highlighted a little bit were to do with the sort of like the use of statistical modeling here. And that's probably where you're sort of thinking about using these sorts of software becomes really important. You're able to sort of like layer up your GIS in such a way that you're trying to answer a particular statistical problem to do with you're answering a particular problem using statistical modeling rather than sort of like this sort of population level modeling that we've been talking about. Lots of uncertainty. There's always lots of uncertainty in these sorts of, and you highlight invasive species as one, but there are many, and we've just emphasised that. I think it's also a sort of like highlights a point that's also made in the chat about the use of ensemble modelling. The idea that actually we might not understand, we can't ever understand the system perfectly. We'll never do that. Again, it comes back to emphasizing that models are representations, they're caricatures of our of our understanding of nature. We've built them into to capture the right biological, ecological, environmental information that's pertinent to the problem that we're trying to address. And then that ensemble modeling just runs, multiple iterations of that. It's like weather forecasting. We would never expect weather forecasting to be at all useful if we were just using a single model. We should have the same sort of challenge against any of these sorts of model, any of this sort of aspects of understanding the outcomes when we're doing control. That actually building models that have different initial conditions, different parameterizations, allow those birth, death, dispersal processes to vary across time and space, and then looking to see what are the consistent outcomes we see gives us then a pretty robust way, to understand those outcomes. So I think it's a really good question. There's lots that to still unravel with this one.
Yeah, 2 quick points on this. I think GIS has an essential role in the historical statistical understanding of the receiving environment. So that historical use is important. I think as a projection, it's much less important because we are not predicting an exact outcome in an exact place at an exact time. If what we are looking for is to say probabilistically for a typical environment at a transition or equilibrium stage, is there harm that we don't like? And I think that the GIS is not really so relevant for that because GIS is about specific places and specific times.
Thanks, John. The next question, actually, the next couple of related questions is from Michael Eckerstorfer. He says, is the ultimate outcome in all cases a stable equilibrium? Some scenarios would rather predict wave-like patterns or fluctuations over time. And relatedly, may the high complexity of the things to model, such as large scale or long time, be more of an important factor than the overall novelty?
Okay, thanks, Michael. That is it's certainly a good point. Stability is a relative term. And I think we illustrated in the example of the North Burton Godfrey model from Burkina Faso that there you're looking at what appear to be relatively stable cycles, but with a 100-fold amplitude over the course of seasons. So you can get quite a lot of variability within, from a global point, a relatively stable equilibrium. And as we pointed out there, at the top of the cycle, you've got one risk, and at the bottom of the cycle, you've got a different risk. And in a sense, I guess it doesn't really matter too much what the scale of the amplitude is, or indeed if it's chaotic. As long as you know where the upper limits and the lower limits are, you can see the transition from one type of risk to another type of risk. And it's at the extremes of the ups and downs in those cycles within the overall stability of an equilibrium that's going to be important.
Yeah, I agree. I think in many ways you're it goes back to what I was just saying about ensemble modeling in some sense, and being able to capture that uncertainty in such a way that you are not, you're not thinking about this necessarily as a point equilibrium in time or space. That actually it's a transition from pre-control to post-control, and understanding how that risk profile changes over that time, rather than the precise dynamics. We've used that sort of language through the talk about thinking about equilibria, but it's just like a state. It's a state pre-control, transition state, post-control state. We've shown that those you can get to equilibrium. One of the illustrations I showed yes, there's a black mass of things around some steady state, but actually there's lots of fluctuations around that steady state. Which would be deterministic, but actually understanding capture within that is all the sort of cycle wave-like patterns that were illustrated by ACE's model at looking at this. May the high complexity of things to model be more of an important factor than the overall novelty? I'm not sure I quite understand what that overall novelty means. I'll take my interpretation. I don't think that, we think about things in time and space. I think, testament to this is the Burkina Faso model that ACE and colleagues have built, that actually is huge. We're thinking about 1 million square kilometers. That actually understanding Anopheles population dynamics on that scale is mind blowing. It's like thinking about outer space in some sense in ecological modeling terms. So actually, I don't think that again, it's predicated on having plausible pathways to harm, understanding this, in this case, the efficacy harm link, and formulating a framework that allows you to evaluate that. I don't think that precludes in any way the outcome of building complex models that allows us to get informative detail out that helps us construct our risk assessments.
Thanks, Mike. The next question comes from Raul Medina. He asks if the local extinction of a target species controlled with gene drives has serious effects on a non-target species that was not picked as a species to be considered in the problem formulation phase of the ecological risk assessment for this gene drive pest suppression effort. A model of gene drive will be unable to predict this ecological impact. So how can one be sure that one is selecting relevant non-target species to monitor or model in systems like tropical rainforests, in which we don't even know all the players involved in the dynamics of those complex ecosystems?
Yeah, that's certainly a very good point. And we have to remember what the role of models are or is, and what else needs to be considered. So we emphasize that models are part of a weighted evidence approach in a risk assessment. So you model what you model, and the models need to be informed by evidence from direct observation and evidence from past published data and other information. They also need to be informed by public consultation, and these other inputs into the overall risk assessment are essential in order to cover the range of species that we should be looking at. But we can only look at species that come to our attention as being of concern. So that indeed is a limitation in the whole process.
Yeah, I see that as not necessarily as not a problem of the modeling per se, but that it's something that we need to take into consideration in the problem formulation phase.
Indeed, models will model whatever is asked of them, and if society asks for particular species or particular examples of species or types of species or conditions of environment to be considered, those would go into the model or the set of models.
Right, and of course the problem formulation phase is designed to identify which interactions are going to be important, either to protect or of concern to the
What one can do, I think, to slightly generalize this is to introduce not specific environments and specific species into a model, but you can imagine a species of a type with properties that are of a particular interest to you, or an environment that might have a particular interest to you. Not necessarily a real one, but if you've got a type of species, a type of target organism and a non-target organism and receiving environment, you can say, well, we don't know about a species that we don't know about, but we understand types of species. So if the species eventually that becomes of interest is of a type that has been included, then we would at least know something about it in advance. So there are certainly some ways of dealing with this.
Okay, the next one is actually from Dave Obrochta, and this question also relates to a previous question he had on ensemble modeling, which you, I think, Mike addressed very well. But Dave does ask again, regarding uncertainty, has there been enough modeling that enables you to begin to appreciate between model variation? For example, modeler 1 finds X and modeler 2 finds Y. Are different modeling approaches leading to different outcomes?
Yeah, so good question, Dave. It's about weather again. So in many ways, I think that ensemble modeling for weather forecasting deals with this in some sense, right? That actually we can deal with uncertainty. We can deal with different, between model variation in the sense that we can average over that. We can think about running, multiple ensembles of different models to get us to a point where we can do pretty robust on a short-term basis weather forecasting. I see no issue in translating that philosophy over to what we're thinking about in terms of releasing gene drive. In that sense, you can build models that give you a way in which to characterize that uncertainty in robust ways, and gives you some probabilistic outcome in the sense that yes, this is going to your transition is going to happen over this time frame. You're going to get down to this sort of broad level of outcome. I think again, asking the precision out of this one is not going to be a good question. It's asking in terms of probabilistic outcome that actually this is what we're expecting to see. I think I think there's still lots to be done here. I don't think in answer to your question, has there been enough work on this yet? No. Is that something that should be done? Yes. Will this sort of like consistency of models that we talked about help with that? Yes. So all of that is, work to be done at some rapid pace, I guess, over the next few years to actually get us to a point where we can appreciate the use of these sorts of frameworks in our risk assessments.
Yeah, my response to this would be first to say if several models are giving different answers, is the difference important? Are they so radically different that it makes a difference to our decision? So that would be one question to ask. And especially if they are different enough to cause us to reject or approve a product, I think that we would want to look at the structure of those models and the inputs in them, the assumptions in them. We'd expect them to be transparent enough that we could understand why the models are giving such radically different outputs. And in that investigation, which I think would need to have some broad peer-reviewed approach, I would expect an understanding to come out as to which one gave a better representation of reality.